Hi everyone, welcome back to A Level Biology Help. So in this video I will be taking you through the third required practical for AQA A Level Biology, which is the production of a dilution series of a solute to produce a calibration curve with which to identify the water potential of plant tissue. At the end of the video I will also be going through an exam question related to this practical, as in the written exam the examiner can ask you questions related to the practicals. Right, so let's get on to it. So first we need to identify what is a calibration curve, as you probably will not have come across one in the past. So a calibration curve is a graph that is used to find an unknown concentration of a substance. And to do this, we can compare to a set of samples of known concentrations. So here I have an example of just a random calibration curve that I've found on the internet. Now, as you can see, on the x-axis we have a set of concentrations, and in this particular experiment they have measured the instrument response in response to the concentration. Now, to find an unknown concentration in this case, we would measure the instrument response of the substance with the unknown concentration and draw a line to the line of best fit and then follow down to the x-axis to find its concentration. In this case it is approximately 0.85 milligrams per litre. But in this practical we use a calibration curve to find the water potential in potato tissue. So now we're going to talk about what a dilution series is, as you probably will not have been taught how to do dilutions in the past. So in order to make the collection of samples with known concentrations to do the calibration curve, a dilution series can be employed. So what we would do to get this known concentrations is we would, for example, if I just draw this, have a beaker or whatever with, say, 1 mole per decimeter cubed glucose solution, for example. And we would make a dilution series. And each time the glucose solution will get more and more dilute. So for example, we can add a certain volume of glucose into here and top it up with water to make, say, 0.8 moles per decimeter cubed, then 0.6 and 0.4 or 0.2. It really depends on what values you're needed to do for the practical. So they get progressively more dilute. That is what a dilution series is. So now I'm just going to give you a bit of background into the practical, so talking about osmosis and water potential. I'm only going to go into this briefly, so if you want to learn more about this in more detail, please check out my transport across cell membranes video, which should be linked in the corner. So osmosis, as you will probably know from GCSE, is a movement of water, let me just underline that, across selectively permeable membranes from a high to a low water potential. Now, the term water potential might be new to you. The water potential, to me, is basically like a fancy term for water concentration. So if something has a high water potential, it will most likely have a high concentration of water. So it will be more likely to, the water molecules will be more likely to move across to a lower water concentration. Yeah, so to sum that up, the water potential is the relative tendency of water to move from one place to another. So if something has a high water potential, as I just said, it will be more likely to move. And this is determined by the solute concentration, so the amount of water or sugars. So if something has a higher solute concentration, so a solute would be something like glucose or a sugar, it will have a lower water potential. So water will be more likely to move into it. So now let's get on to the actual practical. So the first step is to do your dilution series. And in this particular example, you make a dilution series of 1 molar sucrose solution. 1 molar is the same as 1 mole. Oops. Just cross that out. 1 mole per decimeter cubed. I just prefer to write it as a capital M or molar because it's just easier to write, really. And what we're going to do in this dilution series is make concentrations of 0 molar, 0.2, 0.4, 0.6, 0.8 and just your 1 molar solution. And to do these dilutions, we're going to make them up with distilled water. And we're going to make 20 centimetres cubed, so 20 ml of each of these solutions. So how do we know how much water or how much of the 1 molar sucrose solution that we're going to put in? So this is a table that shows you how you do this. You might already be provided with a table like this by your teacher, or you may even be required to calculate it yourself. But it's important to learn how to do it because the examiner might ask you to calculate a dilution series in the exam. So for example, for the 0 molar solution, obviously you don't need any sucrose because it's 0 molar, so you just do 20 centimetres cubed of distilled water. So for the 0.2 molar, for example, this is how you calculate that you need 4 ml of the sucrose solution and 16 ml of the distilled water. So what we do here is we calculate a dilution factor, which tells you how much a solution has been diluted by. And to do this, we divide the old concentration, so 1, by 0.2. And if you have calculated it already, that will come to a total of 5. So we have a times 5 dilution. Now the next step is to calculate how much of the solute solution, so the sucrose solution, you need to make a 0.2 molar solution. So to do this, you divide the final volume, so that's 20 in our case, by the dilution factor, which equals 4. So this means you need 4 ml of the 0.2 of the 1 molar sucrose solution to make your 0.2 molar solution dilution. And then to calculate the volume of distilled water needed, you just take away the 4 from the total volume. So you need 16 ml to make your 0.2 molar sucrose solution. The next step is to use a cork borer, which is like a metal tube, to cut 6 identically sized chips from the potato. It's really important here that you make them identically sized so that the experiment is reliable, because obviously if you have a larger chip then it will hold more water and that would make the results not accurate. So you cut the chips from the cork borer by using a scalpel, obviously. And then you blot to remove excess water. The term excess is really important here because you don't want to rub the potato too dry because that will remove some of the water. Then you have to weigh each chip, which is really important here because the point of the experiment is to calculate the percentage change in mass of each chip, so to figure out how much osmosis has happened. So after you have weighed each chip, you place 1 chip in each of the solutions, so the 1 molar, 0.8, 0.6 and so on solutions, and you leave them for 20 minutes. You then remove them from the solutions, briefly blot them to remove the excess, and then you calculate the percentage change in mass of each chip. So you might be wondering, well, I don't really know how to calculate percentage change in mass. So to do this, you have your original mass. So for example, if it was, it's just an example, but if it was 10 grams, then if your final mass was, say, 5 grams, you do it like this. So you subtract the new mass away from the old. You then divide by the old mass, so that's 10, and you multiply by 100. And that will come to a total of 50%. And obviously, because the mass has decreased, we would say it is a 50% decrease in mass. So now you have calculated your percentage change in mass, you need to produce your calibration curve so that you can find the water potential of the potato tissue. So in order to produce a calibration curve, you plot a graph as a change in mass, so your percentage change in mass, against the concentration of the sucrose solution that you have made. And it will probably look something like this. So you plot your sucrose concentration, so let's say 1. This is just a rough sketch, it's probably not what your results will look like in class. And let's say -10, +10, +20, and so on. So these are not accurate numbers, it's just to show you what the graph should look like. And the important thing here, to find the water potential, is to look at where the line of best fit crosses the x-axis, so here. And this is where your solution is, so this is where your solution is isotonic. Well, let's cross that out, pen's not behaving very well today. So that is where the solution is isotonic, so there is no net movement of water. So the water potential here, at this point where the line crosses the x-axis, represents the water potential of the sucrose solution, which will be the same as the potato tissue, as there is no net movement of water here, because it crosses the x-axis, where, as we can see, there is a 0% change in mass. So to find the water potential of the potato tissue, you have to look online to see what the water potential of so from this graph, for example, a 0.8 mole of sucrose solution is. Once you have that number, you can assume that that is the same as the water potential of the potato tissue. So that is how you interpret your results. So this is just a brief slide to show the risk assessment. So the hazards are broken glass and scalpel. They both have the same risk, so you can get cuts because they are sharp objects. For broken glass, so with your test tubes, you take care when handling and you keep them away from the edge of the bench because you don't want them to fall off and you don't want to hurt yourself. For the scalpel, you can use forceps to stabilise the potato when cutting, keep them away from the edge of the bench, and cut away from your body so you don't accidentally cut yourself. So now I'm going to go through the expected results and the conclusion that you can take away from this experiment. So in the potato tissue that has been soaked in lower sucrose concentrations, there will be an increase in mass. This is because the potato has a lower water potential than the solution. So there is a higher water potential gradient. The term water potential gradient is quite important in exams because the examiner likes it when you write this term, so I'll just write it here. Yeah, because so the potato has a lower water potential as the sucrose concentration is low, so the sucrose solution will have a higher water potential because there's a higher concentration of water molecules compared to sucrose molecules. On the other hand, the potato that has been in a higher sucrose concentration, so your 1 molar, 0.8 molar, 0.6 molar, will show a decrease in mass. This will be because the potato has a higher water potential, so a higher water concentration than the sucrose solution. This means that there is a water potential gradient that goes from the potato to the solution. So water moves out of the potato tissue by osmosis. Right, so now I'm going to go through an exam question that is based on the theory behind this practical. So, 6 cylinders of a standard size were cut from a single large potato. 1 cylinder was placed in distilled water and the others were placed in sucrose solutions of different concentrations. The length of each cylinder was measured every 5 minutes for the next 50 minutes. The graph shows the changes in length at each sucrose concentration. So the first part of the part A of the question is, explain why the potato cylinder in distilled water increased in length. So as we can see from this fairly complicated looking graph, this is not a calibration curve, by the way. We can see that the line is best fit for the distilled water potato. It increases in mass fairly rapidly. This is because the distilled water has a higher water potential than the potato, as it is just water. So the water moves into the potato tissue by osmosis. So this is what I have written for this question. The potato has a more negative water potential, or you can write a lower water potential, it doesn't really matter. So water enters the potato by osmosis, so the cells extend in length. It is important here that you write about the cells in the potato tissue extending in length, because the question is asking you to explain why the potato cylinder increased in length. So it is wanting you to refer to the length here. So if we take a look at the mark scheme, the first marking point is that the potato has a more negative water potential, or you can write that it is hypertonic, which is a fancy term for having a more negative water potential, but it isn't essential that you know what these terms mean. You can just write a more negative or a lower water potential. Also, the mark scheme accepts more concentrated. So if you put that, you can still get a mark. The second marking point is that water enters by osmosis, so it's important that you write by osmosis here, causing the cells to extend, or you can write become turgid. Now, as the term cells is underlined here, it is crucial that you write cells to get the mark. If you didn't mention cells, then you won't get the mark. Because in osmosis, it is the cells that expand, so to increase the mass. Right, so the second part of part A is to explain why the potato cylinder in the 1 mole per decimeter cubed sucrose solution showed no further decrease in length after 40 minutes. So we need to take a look at the 1 mole per decimeter cubed line, which is this one. So as you can see, it decreases exponentially until it reaches a plateau about here. Oops. So what I've put, all of the water has moved out of the potato because osmosis is a mode of transport, so equilibrium has to be reached eventually. And the cell wall of the potato cells prevents any further shrinkage. Now, this part about the cell wall isn't mentioned in the transport across cell membranes topic, but what is crucial in the exam is you collect knowledge from different units to apply your knowledge to an unknown situation. So here I've used my knowledge from the structure of eukaryotic and prokaryotic cells topic to enhance my knowledge in osmosis. So there is no further shrinkage in the mass, so there is no further decrease in the length. because of the cell wall, because it becomes turgid. So the mark scheme says there is little or no water remaining in the potato. You can also put fully plasmolysed, but you don't need to really remember those terms. Or you can put all the water has moved out. You can put either one of those terms to get the mark. I have put all the water has moved out, so I will get that mark. And the second marking point is the cell wall prevents further shrinkage, or you can put the sucrose solution moves in. Now the reason why the length doesn't increase when the sucrose solution moves in is because all of the water has already moved out, so there is no further difference in the length. So you can put either one of those to get that second mark, and I put the point about the cell wall, so I will get both marks for this question. So now let's get on to part B. So the first part of part B says, describe the difference in the rate of decrease in length during the first 10 minutes, underline this, between cylinder in the 0.4 molar solution and the cylinder in the 0.8 molar solution. So first we need to look at what both of these rates look like. So here's a 0.8 and here's a 0.4. So as you can see in the 0.8 molar rate, the length decreases more rapidly and the 0.4 molar decreases less rapidly. Here's what I have written for this question. In the 0.8 molar solution, there is a faster rate, so it is really important that you mention rate here, of the decrease in length. I don't need to explain anything because it is only a 1 mark question and the question is asking you to describe and not explain. Here's what the mark scheme says. So the mark scheme just says there is a faster rate of decrease in the 0.8 molar solution. You don't actually need to mention decrease because it is in brackets, but it is useful so the examiner understands your answer. So now for the second part of the part B, is asking you to use your knowledge of water potential to explain this difference. So you need to write why there is a faster rate of decrease in the 0.8 molar solution. The 0.8 molar solution is higher, more concentrated than 0.4 obviously, so this means that the 0.8 molar will have a more negative water potential. So here's what I've written. I have written that there is a larger water potential gradient. I recommend that you write about water potential gradients in the exam because it is often a crucial marking point. And sometimes you can only get a mark if you write about the water potential gradient. Obviously, if the water potential difference is more negative, is larger, there will be a larger water potential gradient, so water will move out or move in quicker. So the mark scheme says bigger water potential gradient, I put larger, which means the same thing, so I will get that mark. Or you can put there's a greater difference in water potentials between the potato and surrounding solution, although you don't need to mention this because it is in brackets, although it does make it clearer. Right, so we are now moving on to part C, which is different. So, after 45 minutes, the potato cylinder in the 0.8 molar solution was removed and blue dye was added to this solution. Some of this blue stained solution was drawn into a syringe. A drop was then released, slowly, halfway down a test tube of fresh 0.8 molar sucrose solution as shown in the diagram. The blue drop quickly moved to the surface of the liquid in the test tube. The density of a solution depends on its concentration. The more concentrated the solution, the greater its density. Explain why the blue drop had a lower density and therefore moved up. Right, so as this is an explain question, you need to explain why something is happening. It is useful to always highlight the command words here. So first we need to look at what concentration we have. So we have the 0.8 mole per decimeter cubed or 0.8 molar solution here. And this is quite a concentrated solution. In the question, it says the density depends on the concentration. It also says the more concentrated the solution is, the greater its density. Obviously, this means that the sucrose solution will have a greater density because it's quite concentrated. So this kind of explains why the blue drop had a lower density. So we can figure out that the blue drop had a lower density because it is less concentrated than the 0.8 molar sucrose solution. So I've written the drop had a lower density because it becomes less concentrated. This is because the water moves into the solution from the potato by osmosis. So the water moves into the solution because the potato has a higher water potential than the solution, as the sucrose solution is quite highly concentrated with solute. So yeah, the water moves from the potato moves into the solution, which means that the potato becomes less concentrated in the end. So this decreases its density. So if we look at the mark scheme, it says water has moved into the solution from the potato. I've written that so we can get that first marked. And the second marking point is that because the solution is diluted, / becomes less concentrated. So we will get that second mark. So the second part of this part of the question says the sucrose solution of concentration 0.3 molar has a water potential which is equivalent to that of the potato cells. Describe and explain what would happen to the blue drop from the solution. Now, as this says describe and explain, you don't need to just explain what happens, you need to explain why something is happening. So this is what I've written. I have said that the drops would not move, so nothing would happen to the drops, as there would be no net movement of the water. You need to refer to net movement, not just movement, because the definition of osmosis is a net movement of water. So there is no net movement of water here because the solution used here of 0.3 molar has a water potential that is equivalent to that of the potato cells. So there is no room for water to move, so the solution is isotonic. So if we look at the mark scheme, there is no net movement of water in or out. You don't need to write in or out because it's in brackets. So the drops move up or they become less dense. Or you can write there is no net movement of water, so the drop would not move or the density stay the same. I have mentioned that the drops would not move, so I would get both marks here. Now notice that the term or here is underlined. This means that you can only write one of these two sets of marking points. If you write all four, you wouldn't get any extra credit because it is only a 2-mark question. Right, that is all I have to say for this video. Thank you very much for watching. If you have any video requests or questions, please leave them in the comments. Thank you so much for watching and goodbye.